Hello, my name is Jessica, and I'm a Senior Field Application Scientist at Oxford Nanopore Technologies. In this masterclass, we will be discussing how to select the right library preparation workflow for your experiment. In this masterclass series, we will guide you through the Oxford Nanopore workflow from sample preparation to data analysis, so that whether you're new to Oxford Nanopore sequencing or are experienced with the technology, you can learn more to support your application. Now let's continue your preparation journey by reviewing how to select the right library prep workflow for your experiment. In this masterclass, we will review the range of library preparation solutions available to directly sequence DNA and RNA and investigate cDNA. The different Oxford Nanopore sequencing techniques available, including PCR-free and targeted approaches, and how to select the right sequencing kit for your research application. Let's start with an introduction. A comprehensive range of library prep kits are available to support your research goals. We offer solutions for the sequencing of any fragment length, from short to ultra-long, for sample types across native DNA or RNA and cDNA, for whole genome or targeted techniques. Our library preparation solutions offer all-around benefits, with simple, rapid workflows and barcoding options for multiplex sequencing. Fragmentation is not required for nanopore sequencing, and neither is PCR, allowing the direct detection of methylation. Oxford Nanopore sequencing is highly amenable to automation on robotic liquid handlers, enabling hands-off, reproducible preparation of sequencing libraries. In this masterclass, we are focusing on the library preparation step of the Oxford Nanopore workflow, but we also offer support from sample to answer. In our application workflows, we offer complete end-to-end guidance in step-by-step protocols. These include protocols for multi-omic human variation sequencing, single-cell transcriptomics, telomere-to-telomere assembly, microbial isolate sequencing, rapid metagenomics for pathogen surveillance, and complete plasmid analysis. As well as offering manual library preparation methods, our teams also offer support for automated solutions. Our manual library preparation is simple, making the transition to automation straightforward. Our protocols are compatible with a range of liquid handlers and include multiplexing options, with the resulting prepared libraries being compatible with our range of scalable sequencing devices. Our knowledgeable automation solutions team is available to assist with your transition into automation of library preparation. Incorporating automation can be beneficial for other steps in your workflow as well, such as extraction, normalization, fragmentation, and pooling. With this in mind, our teams have developed Ligation. This benchtop device does not simply serve as a liquid handler, but rather an end-to-end automated Oxford Nanopore sequencing solution. The easy-to-use device minimizes hands-on time and does not require prior genomic expertise, enabling efficient automated workflows across a range of applications. Ligation reduces turnaround time to deliver answers when they are needed. The following animation demonstrates how Oxford Nanopore sequencing works. For a more detailed introduction to this process, we recommend you watch the first masterclass in this series, sharing how to get started with Oxford Nanopore sequencing. Here, let's focus on the steps needed to prepare a library for this process. A key part of Oxford Nanopore library preparation is the addition of sequencing adapters to the ends of DNA or RNA molecules. A motor enzyme is coupled with these adapters, and it is this motor enzyme that feeds a single strand of DNA or RNA through the nanopore, as you can see here. The motor enzyme also acts as a molecular brake to slow down the translocation speed of the molecule. As the molecule translocates through, each component will cause a distinctive disruption of the ionic current passing through the channel, producing the electrical signal, otherwise known as raw squiggle data. Understanding this mechanism is key to further unlocking the potential applications of working with this sequencing data, particularly when sequencing native molecules. As we are able to sequence DNA and RNA natively, the data generated does not include PCR amplification bias, and with no additional preparation steps, enables modified base detection. And as the technology is read-length agnostic, We can sequence strands from as little as 50 bases to millions of bases in length, all in real time. Now, let's take a closer look at the process of preparing sequencing libraries, starting with DNA samples. Oxford Nanopore Technologies offers two main chemistries for sequencing native DNA, ligation-based and rapid chemistry, with the ligation approach optimized for maximum output and the rapid chemistry optimized for speed. Many areas of the genome are inaccessible to sequencing by amplification-based approaches. PCR prefers GC-neutral areas of DNA and preferentially amplifies shorter DNA fragments. While this is fine for targeted approaches, when examining a genome via amplification, these factors result in longer or GC-rich PCR templates amplifying less efficiently, or not at all. As an additional point, any amplification-based approach will lose epigenetic markers as these are not preserved through PCR. In order to get as unbiased a representation of a genome as possible, sequencing the actual native DNA is required. Native PCR-free nanopore sequencing methods are required in order to directly detect methylation, avoid PCR bias, and provide more control over read length. Now, let's take a closer look at how these PCR-free library prep methods work. The ligation chemistry is our most popular option, which is no surprise given its versatility and high outputs. While it is optimized to deliver the highest output of any of the kits, the library prep is simple and straightforward to perform. The first of two enzymatic steps repairs damage and nicks in the template molecules and prepares the fragment ends for ligation. This means blunting overhangs and generating uniform ends with 5' phosphates and 3' adenine overhangs. The second enzymatic step is the ligation of the sequencing adapters, which feature thymine tails that are complementary to the DA-tailed templates. The whole process, including the cleanups after each enzymatic step, takes around 60 minutes to complete. This process is highly versatile and able to accommodate any sort of double-stranded DNA, including cDNA and amplicons, not just genomic DNA. You can control fragment lengths going into the preparation by optional fragmentation and/or size selection. As covered in detail in the Masterclass, How to Extract High-Quality DNA and RNA. The ability to multiplex up to 96 samples without PCR is also available using the native barcoding kit, which has an additional step of barcode ligation to each individual sample prior to pooling of these samples and ligation of the sequencing adapters. The preparation time for native barcoding can extend up to 140 minutes, depending on the number of samples you wish to multiplex. Our rapid sequencing kit is optimal where time to answer is critical, or access to the lab equipment is limiting. In a single, quick step, the transposase-based chemistry randomly fragments the DNA molecules and tags the exposed ends of the fragments with adapter sequences. After this, a sequencing adapter is rapidly attached onto the tagged ends. The whole process only takes around 10 minutes, and no third-party reagents are required. High molecular weight DNA is the ideal input for this kit to account for the random fragmentation by the transposase. This method is also well suited for plasmid DNA sequencing, as the transposase cuts open the circular plasmid, eliminating the need for a separate linearization step during prep. The same transposase chemistry is also used in the rapid barcoding kits, which enables multiplexing of up to 96 barcoded samples. Here, barcodes are attached to the DNA molecules by a barcoded transposase complex in the first step of preparation. These barcoded samples are then pooled and spray cleaned prior to attachment of adapters. The preparation time for rapid barcoding takes up to 60 minutes, depending on the number of samples you wish to multiplex. The rapid chemistry has also been adapted for use in the ultra-long DNA sequencing kit, enabling library preparation of ultra-high molecular weight DNA. This method delivers read length in 50s of 50 kb and greater, and reads reaching megabases in length, while preserving base modifications. The ultra-long reads generated are particularly beneficial for assembly, structural variant calling, and phasing. The workflow starts with extraction of ultra-high molecular weight DNA At the start of library prep, a tagmentation step is performed. Here, a high ratio of ultra-high molecular weight DNA to the transposase complex is used, resulting in fewer cuts per molecule and retaining ultra-long fragments. Adapters are then attached to the tagged fragments, followed by a cleanup using precipitation buffer. After the recommended overnight final elution step to homogenize the library, the ultra-long fragments are ready to be loaded on the flow cell for sequencing. For those with a limited amount of input material, PCR-based sequencing kits are available. The PCR expansion can be used to increase sample input prior to the use of the ligation sequencing kit, yielding high outputs from as little as 100 femtomole of starting sample. Alternatively, the PCR barcoding expansion kit can be used to multiplex up to 96 samples with a preparation time of around 140 minutes after PCR. The rapid PCR barcoding kit is also available for where starting inputs are low and time to answer is important. From a low input of 1 to 5 nanograms, this transposase-based method enables barcoding of up to 24 samples and only takes 15 minutes plus PCR. Oxford Nanopore Technologies also offers targeted sequencing solutions for those interested in exploring specific regions of a genome. This includes the end-to-end 16S sequencing workflow, where the 1.5 kb conserved 16S ribosomal RNA gene is targeted for enhanced bacterial identification. From an input of only 10 nanograms, this method enables multiplexing of up to 24 samples and a quick turnaround time, with a 25-minute library preparation step after PCR. While PCR is not necessary for Oxford Nanopore sequencing, protocols are also available for those aiming to utilize a PCR-based targeted approach, where 100 to 200 femtomoles of an amplicon can be used for input with our ligation sequencing kit. A feature unique to Oxford Nanopore Technologies is adaptive sampling, a PCR-free targeted sequencing approach that does not require additional time at the bench. With adaptive sampling, the whole genome DNA library is prepared for sequencing without any amplification or enrichment steps. The target enrichment, or depletion, then takes place entirely during real-time sequencing. For information on how this process works, check out the How to Analyze Your Oxford Nanopore Sequencing Data Masterclass. As this method is PCR-free, it is particularly well suited for sequencing large or difficult to PCR regions, as well as for exploring base modifications. A wide range of library prep solutions are available to suit your DNA sequencing experiment, whether that be whole genome sequencing solutions optimized for high output with control over read length by use of our ligation sequencing kit chemistry, whole genome sequencing solutions for applications where time to answer is critical and access to lab supplies are limited by use of our rapid kit chemistry, or targeted sequencing approaches, or our computational PCR-free approach with adaptive sampling. As with DNA, there are multiple approaches for those interested in RNA sequencing. The direct RNA sequencing kit is our library prep solution for the preparation of native RNA samples. This technique enables the analysis of full-length transcripts, poly-A tails, and discovery of RNA modification. The simple 135-minute library prep method begins with conversion of a single-stranded RNA to double strands at the 3' end by annealing a splint to poly-A tail. An optional reverse transcription step is then used to iron out the kinks and secondary structure present in RNA templates, which can lead to blocking and reduce yields during sequencing if not straightened out. Finally, a sequencing adapter featuring the motor enzyme is attached. In sequencing, only the native RNA strand translocates through the nanopores and is sequenced, even in the presence of the reverse transcribed strand. Those looking for higher data outputs and the option to multiplex will find the cDNA sequencing workflow an ideal solution, with data enabling the characterization of isoforms, splice variants, and fusion transcripts, as well as isoform-level expression analysis, poly-A tail length estimation, and the ability to identify and quantify full-length transcripts. Library preparation begins by annealing a reverse transcribed adapter to the poly A tail, followed by first-strand cDNA synthesis. Strand switching is then performed with incorporation of unique molecular identifiers and followed by PCR amplification with rapid attachment primers that enable attachment of sequencing adapters post-PCR. This preparation can be completed in 225 minutes, along with the time for PCR. A barcoding kit is available that enables multiplexing up to 24 cDNA samples. Sequencing methods are also available for those interested in sequencing cDNA at single-cell resolution. Single-cell cDNA prepared with 10x genomics kits can be used for Oxford Nanopore sequencing with our single-cell protocols, which utilize the ligation sequencing kit. This application supports analysis of full-length transcripts to reveal isoform diversity and alternative splicing, along with identification of variants and gene fusions, all with the streamlined 3-hour library preparation protocol. With long Oxford Nanopore reads, you can capture full-length transcripts, revealing a more complete picture of isoform diversity. Not only are you able to explore epigenetic modifications, but also full splice information, differential gene expression, fusion genes, and more. We look forward to supporting your application by guiding you to the best solution for RNA sequencing, even up to single-cell resolution, with options available for different input quantities, multiplexing needs, and analysis goals. Now, let's take a look at a worked example of an Oxford Nanopore sequencing workflow, focusing on 16S sequencing. Targeted sequencing of the 16S ribosomal RNA gene is the predominant method used for the identification of microbes across applications including food safety analysis, environmental and conservation monitoring, and pathogen detection. However, 16S sequencing using legacy short-read technology provides limited resolution. As short reads cannot span the entire 16S gene, only partial fragments are sequenced instead, making species-level identification challenging. In contrast, the full-length 1.5 kb 16S gene can be captured in single Oxford Nanopore reads, delivering high taxonomic resolution for accurate species-level identification. For the 16S workflow, libraries are prepared for sequencing with the Oxford Nanopore 16S sequencing kit. Using this kit, the entire 16S gene is amplified, and barcodes are added for sequencing of up to 24 samples in multiplex. Using rapid attachment chemistry, the sequencing adapter attachment step takes just 10 minutes. Now that we've seen the library prep step, here's the end-to-end workflow. From recommended extraction techniques for your sample type, to full-length 16S sequencing on the portable MinION for up to 24 samples on one flow cell, to data analysis with the intuitive 16S epitome workflow without the need for prior bioinformatics experience, we've got you covered from sample to answer. Let's take a look at the end-to-end direct RNA sequencing workflow as an example. Modifications in RNA play a significant role in the progression of cancers and neurological disorders. However, native RNA cannot be sequenced using legacy sequencing technologies that require conversion of RNA to cDNA, meaning that these modifications are lost. With Oxford Nanopore direct RNA sequencing, it is possible to sequence native RNA, not only capturing full-length isoforms and allowing poly A tail measurement, but also ensuring that RNA modifications are preserved and can be detected. When it comes to library preparation for this workflow, we offer the direct RNA sequencing kit. This PCR-free method prepares native RNA for direct sequencing, preserving long fragments and modifications. The reverse transcription is optional, and the reverse transcribed strand is never sequenced. Only the native RNA strand passes through the nanopore. Here's the direct RNA sequencing workflow from sample to answer. When starting from a human cell line, for example, we recommend extracting RNA using Trizol RNA isolation reagent, then enriching for total RNA with the Invitrogen Dinyl Dynabeads mRNA purification kit. After library preparation, we recommend sequencing a human transcriptome on PromethION flow cell for high outputs of full-length transcripts. Finally, Transcriptome's Epitome workflow provides intuitive transcriptomic analysis. We invite you to learn more from our online resources, where we provide tools to aid you in getting started and continuing to grow with the technology. Featuring training videos, step-by-step protocols, and end-to-end workflows, our website will aid you in identifying the ideal library preparation method to suit your needs. Follow the QR codes here to find out more. In this masterclass, we reviewed how to choose the right library preparation kit for your application and experiment goals, with options available to enable native sequencing of DNA and RNA, straightforward single-cell and bulk transcriptome sequencing, and PCR-based and computational targeted sequencing approaches. We also encourage you to review the wealth of resources available online. Thank you for joining this masterclass on how to select the right library preparation workflow for your experiment. We look forward to seeing you in the following masterclasses, where we will continue to take you from sample to answer.